Good evening, everyone. It's my immense pleasure to host this meeting. I am T. Prenya. As we reach the final day of this webinar series, it's a moment filled with both accomplishment and gratitude. Over the past sessions, we have explored new ideas, gained valuable insights, and engaged in meaningful discussions. This journey would not have been the same without the active participation, dedication, and collaborative spirit of everyone involved. As we move towards the conclusion of today's webinar, we have the pleasure of hearing from someone who has truly reflected the purposes and values of this program, Jignasa. Jignasa has been a consistent and committed presence throughout this webinar. It has shown enthusiasm for learning, clarity in understanding, and a positive approach towards every task and discussions. Their willingness to engage thoroughly and contribute meaningfully has added great value to this experience. What set Jignasa is apart is the calm, confidence, and sincerity. It approaches every opportunity with focus and responsibility, and the efforts demonstrate not just knowledge but also professionalism and teamwork. These qualities are essential in today's learning and working environment, and Jignasa has displayed them gracefully. On this last day, it is fitting that we hear from us if we reflect on the journey of this webinar and look ahead to applying what we have learned. The words will help us bring the session to a meaningful close and reinforce the key takeaways of our time together. It is the last but not the least. I would like to thank all the volunteers of Jignasa team for your time and dedication. We could not have done it without you all. Thank you.
நந்தி அகத்தியர் மூலர் உண்ணாக்கீசர் நற்றவர்க்கு புலத்தியரும் பூனை கண்ணர் நந்தியை காணரும் போகர் புலிக்கை ஈசர் கருவூரார் உங்கணவர் தாலாஞ்சி சிந்தி எடுக்கண்ணர் அகப்பேயர் பாம்பாட்டி தேரையரும் குதம்பயரும் சட்டைநாதர் செந்தமிழ் சேசித்தன் பதினெண்பர் பாதம் சிந்தையுன்னி சிரத்தணியார் சேத்தி வாழ்வார்.
Today, we are honored to have Dr. S. Sudha Revati, MDS, Postgraduate Diploma in Herbal Biotechnology, PhD, as our guest speaker. She is an assistant professor in the Department of Gunapadam at the National Institute of Siddha, Chennai. She is a respected Siddha physician and researcher. Dr. Sudha Revati has completed her BSMS from Government Siddha Medical College in 1998. She earned her MDS in 2002, PG Diploma in Herbal Biotechnology in 2005, and PhD in Siddha in 2016 from the Tamil Nadu Dr. MGR Medical University. Her work focuses on preclinical evaluations, pharmacovigilance, and herbo-mineral formulations. Her research includes key publications such as the 2024 study on in vitro study of Vepampu, oral kudineer for anti-ulcer activity, and her PhD thesis on Karasuda Sattuparpam for urolithiasis. She has published over 20 peer-reviewed papers on topics like urolithiatic activity, standardization of Siddha drugs, and Siddha treatments for liver diseases, iron deficiency anemia, and computer vision syndrome. She has also co-organized the national seminar on recent researches in Siddha in March 2022. She served as co-investigator on Ministry of Ayush projects for anemia in adolescents and oropharyngeal cancer management. She has been a resource person in CMEs on herbal standardization and GMP for Ayush drugs. Her skills cover toxicity studies, antitoxin screening, and quality control under ISO standards. Let us welcome Dr. Sudha Revati and invite her to start today's session on standardization of Siddha drugs. Welcome, ma'am.
Good evening, one and all. Am I audible to you all?
Yes, ma'am, you are audible.
Yeah. As the student who has mentioned about me, I'm Dr. Sudha Revati, presently working as Assistant Professor in Department of Gunapadam in IS Chennai. And the topic which I have chosen today to speak is on standardization of Siddha drugs, which is a part of Gunapadam and also I have started my journey in that standardization and testing of Siddha drugs as my career, and it is very much overwhelming to see the young students, especially the UG Siddha students, who has taken part in Jignasa as part of promotive activity of Siddha. I heard this from one of my student, Shivaharshini, and she told about these activities. And she only has suggested my name, it seems. And after that, Hariharan has followed me on this. And readily I have given consent. And I was happy also to see such a voluntary thing happening from the student side. And this will definitely create a lot of awareness on Siddha among the public, and also I think you have to go a long way on this. I wish all the students who have been part of this Jignasa activity in southern part, and I congratulate all the students for actively participating in this. So with this, and also I thank my director who has permitted me to conduct this session on behalf of NIS. Now we'll move on to the topic directly. First slide, please. Next slide, please. So the topic is on standardization of Siddha drugs, and now we'll know like what a standard is. So when we talk on standardization, it's a huge topic. And what is a standard? Standard is nothing but it's a numerical value. It should have a value, and it should be quantifiable. What does it say? If you're going to quantify something, what should it reflect is the quality of anything. Here, it should reflect the quality of the drug, purity of the material, and simultaneously, what is going to happen when you have a standard with a numerical value which is quantifiable and that is going to reflect your quality? What does it reflect on? Of course, the efficacy of the drug. So, the important thing is whatever we call a standard is, it should possess a numerical value and it should be quantifiable by some parameter that is going to reflect on the quality and ultimately on the efficacy of the drug. So this is what a standard is. Next slide. Now, my core topic is standardization. So now we know what a standard is. Now we'll see like what is a standardization. So what is a standard is a numerical value and it's a parameter. Now what is a standardization? It's a process. What is it going to do? Now you know what a standard is. So by this process, what is that we are going to achieve is a set of standard. It is not one standard for one medicine or one raw material. It is not like that. It should be a set of process or set of standards. So what it should contain? It should give a characteristic picture on that particular product or the raw material. And the most important is it should be a constant parameter. So whenever you do that, the quantitative test or something, it should show the same value with the deviation which you have set. And it should be a definite qualitative or quantitative values. And what it should reflect on, again, the quality, efficacy, safety, and reproducibility. So this entire process where you're going to set in standards, it may be one or two or many. That depends on the raw material or the product. So this is a process where reproducibility is very, very important, and ultimately that is going to safeguard your quality of the drug, efficacy and safety of the drug. So it is not that it is a continuous process. Once if you fix a standard, it is not going to be the constant. So sometime after 5 years or something, it may vary. Means depending on the product or if there is any change in the seasonal conditions or something, if there is an updation, it should be updated as per the changes that are happening with respect to the raw material or the product. So standardization is again a process with a set of standards, and it should be a constant parameter that is going to reflect on your quality, efficacy, safety, and reproducibility. Next slide. So we talk a lot on standardization, and why is that it is needed? Is one is don't go even for global acceptance. See, we have so many companies within ourselves, ranging from many private, there are semi-government organizations, there are NGOs who are manufacturing the products. So what happens is we have to have an acceptance among ourselves. Like one product, let it be even a simple single drug or a polyherbal combination, or let it be a herbal or herbomineral, whatever it is. We should have our own acceptance values, like yield or how it should appear, how the taste should be, whatever it is. So even for us to accept it to be a drug or accept it to be a raw material, we need some values to be evaluated. For that, we need standardization. Again, if we have our own set of standards or some specification that is generated within India, and if we can easily go in for a global acceptance where the foreign countries are going to give you the guidelines, and if we need to market our products outside, then we can easily just go upon to upgrade our standards to international level. So this process of standardization will help us for the global acceptance. And in the process of standardization, what do we do actually? How do we achieve it? Is that we have to document whatever is the process. Let it be your first batch is going to say it is going to show you one value that should be documented. And after 6 months, again you are going to manufacture the same product and you are going to check whether it has the same values or whatever acceptance values we are given. Is there any deviation or something? So every time you will document what you're doing. For that purpose, we need the process of standardization. Again, what does it reflect? It is going to be an evidence for your safety and efficacy profile. So once we go into the detail of what are all the things that we are going to do for the standardization, then we will understand how does it reflect the safety and efficacy of the drug. And next is the reproducibility of the findings. So once you stabilize or standardize the product, then you can easily just once you follow the protocol or the standard operating procedures, you could easily see the reproducibility of the findings. Even if there are any variations, you can also fix up a range saying the deviation should be this much or that much or something. And the next one is industrial scale production. What do you mean by industrial scale production is, see, we whatever that is given in the literature is a small scale production. Let it be one palam or two palam or whatever it is. It will be of a small scale where we'll keep producing our drugs on need basis. But when it comes to, I'm sorry, I have some throat irritation. When it comes to scaling up process, what do you mean by scaling up is, so whatever we do with a small scale, like ஒரு பழம் ரெண்டு பழம், but you want to do it in tons or in kgs. The process, even if you're going to follow the same SOP, there will be variations. That once you do it, you will understand. So you have to standardize a small scale production and also a scaling up. Scaling up means you have to do a 3 batch analysis to improvise your industrial scale. Next is, why do you do standardization is to know the shelf life. Say for example, our Siddhas have said like 3 months shelf life period for a churnam or a butter based drug. What does it happen after that? Either fungal attack will happen or there may be contamination, or if it is going to be an oil, after that shelf life rancidity will be formed. So for that, if you have the standardized parameters, then you will come to know whether the product is in live condition or not. Do the phytochemicals exist in the same level or not? So for that, you need standardization. Next slide. So we also know how much authenticity of the drug is very important. What happens is, due to unavailability or endangered plant list, sometimes it so happens that people have started adultering or using a very wrong species. And when it comes to minerals, like we know, we don't have authentic raw materials to say or to compare. So whatever that is given, it is there only in the literature, and many of the people are working on it. And we need an expertise to say whether the particular mineral which is said in the literature is what we are using. What happens due to lack of experience or something? We may use a wrong mineral species or a wrong plant also sometimes. So that to avoid that, once if you start working on a raw material and you say this is what, then we can fix upon. The next is by doing this we can control adulteration also. And where you can with simple test also you can say whether we are using the correct material or not. This process will avoid contamination in the drug. And what happens if, see, unless you have, there will be a dosage given in the text and which we'll be using in our clinical practice. What is the particular raw material that is present in the dose? And what happens if there is overdosage? So, all those things you can find out when standardization is done. And healthcare providers, when they do by themselves, or if they are going to do run a company or something, unless we fix a standard, then there will be no control over the drug usage. And if we have set standards to a drug or a raw material, we can also study how it interacts with other medicines. Unless we have a standard or a process that is going to define our medicines, we cannot read on the interaction with other medicines also. And the other important key aspect is adverse drug reaction. Now, when we use combination of drugs, it is highly difficult for us to understand which drug has produced the adverse drug reaction. So, if we have some certain parameters for that, say for example, in the literature it has said particular drug will cause particular reaction. So, what is that particular thing that is going to cause the reaction? Or if the drug is not used properly, and if it is not used in a proper adjuvant. So when we are going to add an adjuvant, so what is that is going to happen to the pH? What is that is going to happen to the drug? What does it happen internally inside? So either by Siddha way or by modern aspect, we should have some clear-cut methodologies to do it. For that, the standardization process will help. Next slide. Next slide, please. So this quality control is a part of standardization. There are two different processes, what we call as quality control and quality assurance. Quality control is a fault finding, means you have a prescribed limits. So if you don't get that, then what alternate can be done? So this setting standards, the laboratory which has to be used is called as a quality control measure. It is not like you have a start point and end point. So quality control is a check that is going to happen from the raw material usage till the finished product, until it is used by the patient. So it covers everything, like how a raw material should be, whether the processing is okay or not, if there is any hindrance in the processing, how to overcome those hindrances or how to overcome those difficulties in processing the drug. So all these matters even after finishing the product. So how do you transfer it to a packing material, whether it is going to be safe or not, whether it is not going to because in as far as our drugs are concerned, the packing material also is very, very important. We can't just like that pack one mineral drug in a normal container. So what happens when moisture happens? So all those things has to be considered. Labeling, how it should be sealed and how it should be dispensed. So all these things we need checks at all the points. So that will be covered under quality control. Next slide. As you all know, The Siddha Materia Medica consists of herbs, metals and minerals, animals and its byproducts. We will briefly see like what are all the steps that are needed to standardize as far as herbal drugs are concerned, and what are all the criteria that we have to see on when it comes to herbomineral metals or minerals. Animal products can be classified or it can be categorized under mineral and metal category, where some of the parameters will be the same to animal products also. Now we will move on to the herbal drugs. Next slide. Now we are talking so much on the standardization, importance of standardization. So what are all the steps that we have to keep in mind? Are the step one is the raw material. Of course, it is very, very important. If you don't have a authentic raw material, then we will not end up with a good product. Again, is it enough if you have one good raw material? It is not like that. You should have a very proper processing steps. Unless you don't define the process of making the drug, even if you have an authentic raw material, then the finished product uh, will not be the same which we want. And we should have some SOPs for the final product and some of the testing parameters for the final product too. So the steps are, you should have checks at raw material, the processing of the drug, and the final product. Next slide. So, what is the methodology that we have to adopt? This doesn't hold good only for herbals. Whatever drug we are using, let it be herbal, mineral, or an animal product, the methodology is going to be the same. So, we should have clear cut protocols for collection. Either we have to collect by ourselves, or if you are going to procure the raw drugs, so we should identify some authentic vendors who is going to supply that particular drug. Unless we do a market survey and ensure that the product is an authentic, then we will not end up with a good product. We should have systematic identification processes. Means if you identify by organoleptic, by naked eye examination, it is well and good. And if you have any hindrances or if you have any challenges, then we have to step into the next process. And next comes, we have specific purification methods even for herbal drugs and also for herbomineral drugs and minerals. We should have particular method of purification for that particular product. And again, the method of manufacturing, it differs. Like if you are going to use it for 10 patients or 100 patients in among the clinic, the SOP is something different. But if you're going to manufacture it on large scale, there are many guidelines that you have to follow. And the process of manufacturing itself, it differs. Starting from the production plant, where you're going to procure the materials, how is that you're going to pass, whether we can use the raw material or not. There are checks. So whether you're going to accept the product or whether you're going to reject the product. So all those SOPs, it should be definite and specific. Coming to the finished product. Once you have manufactured a product, then you should have some parameters that is going to define your finished product. Again, we know we have 32 different 32 different forms of drugs, like internal medicines and external medicines. So when it comes to churnam, the definition is something different. When it comes to a tablet, the specifications are different. When it comes to a purpum, it has some different specifications. So based on that, the finished product should have different checks. Again, coming to the period of preparation. This like what happens when you do a purpum, especially a salt-based drug on a winter season. That is not exactly possible at all. When you grind the drug, it
Ma'am,
will be
sorry to
hard.
interrupt, ma'am.
Yeah,
Are you feeling well? Shall we continue or you
yeah,
need
yeah,
some
we can
help?
continue. No issues. We can continue.
Okay, thank you.
Others are okay. So
Yes, ma'am.
again, period of
Everyone is okay, ma'am.
fine, good. Period of preparation is very important. So as far as already it is given in the literature, when you should procure a drug, when you should procure a route. So it has some impact on the phytochemicals, everything. Even if you don't do a systematic study, by experience you will be knowing like what is very important when you collect a route. and what is very important when you do a salt-based preparation, when it should be done, especially during winter, you will find a lot of challenges. So it is period and also the season specific. So even for that, you need a methodology to do it. And again, after doing a very good drug, if you don't preserve it properly, again we are going to lose our drug. And finally, the shelf life, unless we are very, very defined on the shelf life, the whole process of making the drug, then we lose our credentials. Unless we are very, very, like, if you fix, like, how do you fix a shelf life is you make a product, you have some standards for that. For example, if you fix for like padangam, or if you fix like the for padangam, if you fix the particle size as a one of the thing, then it should have the same particle size even after 10 years or whatever shelf life you are fixing onto. For example, when you do a syrup, then what happens is the total sugars that is present in the syrup should be consistent even after 2 years if you give the shelf life to be 2 years. So this is very, very important. Next slide. So what are all the tools you can adopt? Because methodology is something different. Though we have methods, if you don't have, I can keep telling you, you have to create standards. We have to create standards. Or are there any defined standards that is already there? So what are all the tools that you will use? One is the traditional standards. I hope you all accept with it. And there are traditional standards that are given in each textbook. For example, not all purple will be white in color. But what happens when a gold-based purple is done? It has a different color. Murukambu Niram, whatever it is, that is given in the text are the traditional standards. Like saying, like there are some traditional standards saying that purple should float on water. So these are traditional standards. Of course, it is needed. Next comes the standards that are given in the Pharmacopoeia or Bureau of Indian Standards. Though we don't have much wide volumes in Pharmacopoeia, but these are regulatory publications where this will pitch into when you go for licensing or when you do a marketing abroad. So these standards will be quoted everywhere where you have to follow. But we have in Siddha, we have some few volumes of pharmacopoeial standards where they were covered with single herbal drugs only. We'll just move on to like how it is given, what are all the parameters they have included in further slides. And finally, we can also adopt modern analytical tools that will, it may be little advanced for you, but at least by this time, if you know like there are such tools, you can use it when you get your specialization done in future. This is on the tools for standardization. Next. Now we'll see move on to the herbal drugs, like what are all the important specifications that are needed and what are all the standardization parameters that are needed. Next slide. So the first thing is I have been repeatedly talking on authentication. This will be the first class I take for my PG students. So whatever the product you are going to do, if your raw material is not going to be authentic, again authentic. Authenticity means if you say it is Malay Vembe, it should be Malay Vembe. How do you do that? We have some parameters for that. Appearance is there. So taxonomical classification is one method where you can authenticate a herbal drug. And the next one is herbarium voucher specimen. What do you mean by herbarium voucher specimen? Voucher specimen is what we deposit. So now I have worked on a particular drug. I know it is a authentic raw material. Now what I will do is, say for example, National Institute is there. We have a museum attached to our institute. Similarly, there will be at least a library in all the academic institutions. So what we can do is I can generate a specific number. So once you submit a sample of your raw material to the library or the museum, they will generate a specific ID for that. And you can just deposit that in that particular museum. and that ID will carry the details of that particular sample. And for the future researchers or for future academicians, they can take up that watch specimen and they can compare with the same drug which they are working on. That will carry a specific ID and a database will be created. It is being done in all the institutions. This is one method where how you can authenticate. Next is macroscopic method, which is going by naked eye examination. By seeing itself, we can say whether the drug is authentic or not. So if there are two similar drugs, it will be highly difficult for us to say whether the, say for example, if two drugs are white in color, the appearance is going to be the same. And what will we do? We'll go for a microscopic examination. We'll find out if there are any microscopic differences. So this is one method. Then there are some chemical methods. Apart from all these, there'll be some chemicals that are present in your plants. Like for example, alkaloids may be present, tannin may be present. So it can show in a particular TLC pattern. Okay. So those chemical methods we can establish. And finally, if we fail in all these and it is much complicated, then we can enter into the DNA molecular fingerprinting. These are molecular methods which will say on like how is your DNA present in it. Let it be leaf or root or whatever it is. You can develop a fingerprint for a particular plant. And now that DNA fingerprinting is available for all the plants, and for us it will be easier to compare. We can collaborate with an institute. This we can use only for a very complicated drug where the authentication is very much challenging. The mostly adopted thing is a taxonomical authentication, macroscopic and microscopic methods. Hardly we can go for a chemical method if it is definitely needed. Now this is on the authentication. Next. Now this is an overview of standardization of herbal drug. Coming to the physical properties in a drug, then we have to definitely know on the moisture content, ash value, and extractive values. This you will know like once you start doing it and how what are all the important factors that are needed. This will speak on the physical properties of the drug. Coming to the macroscopy, as I told you, how does it appear? What is the color of the drug? What is the color of the final product? What is the order and what is the taste? These forms the macroscopical character of that particular drug. Next is chemical. That's what we're talking on. Chromatography is nothing but it's a technique that is going to speak on the color spectrum of that particular drug. Then heavy metal analysis, that is widely spoken in many areas about our drugs, especially the heavy metals like lead, arsenic, mercury, and cadmium. So we have some limits for that. And the pesticides is one contamination that is happening in herbal drug due to pollution, land pollution, other thing. You could see a varied contamination that is happening in the city side compared to that of the mountainous regions. So that will be one marker. And coming to the biological parameters, and we should be very specific on microbial contamination. So does it contain any microbes by itself? So we'll see on like what are all the microbes that are normally present. Then we need to go for a toxicological standardization, and we should ensure it doesn't cause any toxicity to the humans. And finally, the pharmacological evaluation. Coming to the analytical tools, microscopy, we have simple microscopic studies. You do a section, put it under microscope, see what is the prominent feature in that. And sometimes you can go even for scanning electron microscope and powder microscopy. See, if you don't have the drug in whole, அப்படின்னு சொன்னா, then we can go for a powder microscopy. You make it a powder. Now we have only churnam of one per single drug. Then we can't authenticate with that by macroscopic method or something. We have to definitely go for a powder microscopy. Powder microscopy will tell you how does a powder look like. So you put a pinch of powder under microscope, you can see whether they contain fiber or crystals or whatever it is. So these are the essential parameters that are very much needed in the standardization of herbal drug. These are the basic things that we have to adopt. Next. Now we'll see some examples for that raw drugs, like what all that is available. What parameters that we can use for raw drugs. Next. This is one I have taken from the Ayurvedic Pharmacopoeia Part 1. This is about the plant Tribulus terrestris. See, this is how the pharmacopoeia will look like. It will give the picture of that plant. And it will describe about the plant. How does it habitat look like? And what are all the synonyms? And coming to the regional languages. Next. Next slide. See, I'm not going to read out what are all the essentials. Just you have to get familiarized with what is given in the pharmacopoeia. Then they would have defined how the macroscopy of that particular drug looks like. What does the root look like? Stem look like? Leaf? Then the fruit. Next slide. Now that you're seeing the microscopic features, if you just highlight and see, they will tell what are all the major features. When you keep a root under it, what are all the major features that are seen. Then stem, leaflet, and fruit. Next. See, what we have seen all this time is on the macroscopy and microscopy. Those are very important. Next comes the identity, purity, and strength. What does it say? So once you speak on the foreign matter, for example, if you're going to talk on மிளகு ஆர் கடுக்காய்க்கு வந்து அகநஞ்சுன்னு சொல்கிறோம். ஸோ கடுக்காயை நம்ம வந்து என்ன பண்ணுவோம்னா கொட்டையை எடுத்துகிட்டு தான் நம்ம யூஸ் பண்ணுவோம். பட் வென் யூ பை இட் இன் பல்க், சம்டைம்ஸ் வாட் ஹேப்பன்ஸ் இஸ் அதோட சீடோட சேர்த்து வந்துடும். If you buy 1 kg and if the seed is going to be in that for 10 grams or 20 grams, this is a foreign matter in that. So the value is given as it should not be more than 2%. Not only this, let it be a unnecessary sand particle, whatever it is, everything is considered as foreign matter. So the specification is it should not be more than 2%. Total ash will tell you like what are all the minerals or if what are all the minerals that is present in that particular plant. Coming to the acid insoluble ash, here for that particular drug, total ash should not be more than 70%, 17% in case of Boxura. Acid insoluble ash, it should not be more than 4%. Acid insoluble ash abdinale, we can just directly correlate it to the sand particle or silica present. So these three will define you. These are very simple tests which will not cost you more. So these are taken as pharmacopoeial standards. Next is the alcohol soluble extractant. What is soluble extractant? This will speak on what is the nature of the phytochemical that is present in your drug. So if it contains more of alcohol soluble chemicals in it, then the alcohol extractive will be more. And if it contains more of water soluble extractive components in it, then water soluble extractive will be more. These are fixed. Next, coming to the TLC. TLC is a very, very simple chromatographic technique. What we'll do is we'll take an extract, we'll run and see like how many spots. So it's a very, very preliminary test which will not cost you more. You will learn about the technique once we'll talk particularly on chromatography. So today you get introduced only to the TLC. So these are some of the pharmacopoeial standards that are given in the pharmacopoeia of Ayurveda. And similarly, the same thing is adopted in Siddha pharmacopoeia also. Next. And then comes to the properties and action, which are given in our text. So what is the dose, everything. These are some of the literature things that are added in the pharmacopoeia. Next. Similarly, this is taken from Siddha Pharmacopoeia India. They will give you the picture. Next. Similarly, the synonyms. Next. Your macroscopic description, and then how does the root look in microscopy. Next. Yeah, the same. What is the foreign matter? This is taken from Siddha Pharmacopoeia. And finally, they have added HPLC content. The HPLC என்னன்னா, now I said there will be specific phytochemical. For example, they have estimated vitaferin. Vitaferin is one of the alkaloid that is present in Withania. They have given the specification என்ன mobile phase use பண்ணணும், what is the flow rate. It is an instrumental analysis. So this you can keep as reference when you buy a raw material. And these things can be adopted for importing or export also. Export of raw materials also. Next slide. Here again, the TLC conditions about all the phytochemicals that are present. Next slide. What are all the properties that are said in Siddha literature? Next. What are all the formulation, therapeutic uses, and also the dose? This is taken from Siddha Pharmacopoeia. Next. Similarly, BIS also gives you some standards. These things are taken from the pharmacopoeia only. But now that they have come up, this show you that they have also taken up the work of making standards. Just to show you, I have incorporated these details inside. Next slide. Yeah. The same specification you can go through later once I share the PPT. Next. As I told you, macroscopy, microscopy, these two are very, very essential. And we can also do a powder microscopy. What happens, இதில் உங்களுக்கு powder microscopy ரொம்ப ஹெல்ப்ஃபுல்லாக இருக்கும் அப்படின்னா, like when it is going to be a whole drug, it will be easy for you to identify. When the same drug is given as a powder, so you can't do it by organoleptic characters. Definitely you need a microscope to do it. So this powder microscopy will be very much helpful to define your product. Next. Now I'll tell you, this is a case study. This is taken from a published paper. We know, what is the drug that is given in the picture is Anachimukku. Normally we use it for culinary purposes. We use it for food making, biryani, everything. It's generally called as star anise because of its structure. Next slide. This is also another species, Elysium. Previous. This is a similar drug and Elysium anisetum. It also looks similar. But this is a different species, Japanese star anise, it is called as. What happened? It is adulterated with that Elysium verum. See, the problem is, let it be adulterated, but when it is going to show some toxic symptoms, it becomes a big problem and the efficacy will be hindered. It has caused seizure, vomiting, and rapid eyeball movement in children. So how do we sort this? By macroscopy, both the drug look the same. Next slide. Next slide, please. See, now you can observe. The first picture is a microscopic feature of Elysium verum, and the next one is Elysium anisetum. Entirely, it is different. Can you make out? So when the two drugs are going to be similar, then the microscopy will be helpful for you to identify and understand and what is the culprit. Not only in normal cases, but in abnormal cases also. Why this has happened? So what is the toxic principle that has caused that adverse drug reaction? Next slide. This just only the names. When it comes to herbals, these are very, very important parameters that you have to look into. Loss on drying, of course, that will reflect the moisture. Ash values, as I said, the mineral content. Extractive values, the nature of phytochemicals, whether it is going to be water soluble, alcohol, ether soluble. Sometimes we may have to go in for the melting and boiling point. And if the drug has optical rotation properties, then that can be estimated. Refractive index, viscosity, pesticide residue, microbial contamination, heavy metal. These are very, very essential in case of standardization of herbal drugs. Next. As I was talking on TLC, this chromatographic and spectrometric evaluation or some analytical tools. Of course, you need a specific instrumentation for that. just get introduced to the techniques. One is UV visible spectrophotometry. Now, presently, we are estimating the functional groups, total as a functional group assay, not specific phytoconstituents like total alkaloid content, total tannin content. All those things can be estimated using UV visible spectroscopy. This TLC, HPTLC are thin layer chromatography, high performance thin layer chromatography. In this, HPLC is high performance liquid chromatography. These will tell you on the nature of your phytochemicals. This NMR will tell you how the structure is and all that. So these are some specialized techniques that are very specific to the herbal drugs alone. Next slide. Now I'll show a case study where how the TLC has helped to analyze an adulterant that is present in 3 different curcuma species: Curcuma longa, Curcuma xanthoria, and Curcuma zingiber cashemonar. All the 3 contains curcuminoids, curcumin compounds. But see, when the drug is pulverized, or when it is all the 3, when it is given in powder, it will be highly difficult for you to, even sometimes microscopy also fails. Then we have to go in for chemical methods of identification or finding out the adulterant. This particular study has used TLC method. Next slide. Just go through the TLC fingerprint. See, these are the different profile of different drugs, and what you could observe, they are not the same. The different bands represent different phytochemicals. That is what you can understand from this. So you can easily make out the first 3 lines are the standard marker compounds. Next 3 lanes are the 3 different curcuminoids that is taken from different species, which we have seen in last slide. When you compare, you could see the difference in the curcuminoid. So by the fingerprint, you can understand there is, but all the 3 are being adulterated. In the last one, which is called as Java turmeric, it is highly costly. So when the normal ones are adulterated in that, then the purchase or the customer who is going to buy you, definitely it will hinder with its quality. Next slide. Now, we'll rapidly move on to the mineral standards. Hardly another 5-10 minutes, I'll wind off. Next. See, 4 aspects we can look into when it comes to minerals. Definitely, the traditional standards will help us to identify or define our final product. Of course, we have 1 or 2 volumes that is going to speak on the minerals, especially in Ayurveda only. We have volume 7 and part 2 that speaks on the raw materials, especially the minerals. Here, the physico-chemical properties of minerals are very, very important, and we have some different modern analytical tools for minerals. Next. So, what are all the traditional standards? This, I think, we have discussed. Murkampu Niram of the final product. Aya Chenduram, Aya Kanda Chenduram, Aya Vira Chenduram are the Niram. So, based on the color of the final product, we can say whether the drug is finished or not. And as I have already discussed, when it comes to purpum, that is based on minerals or metals, it should be lusterless. It should be fine enough to enter the crevices of finger. It should float on water. It should be smokeless. It should be tasteless, and it is irreversible. Irreversible means whatever way you prepare the drug, even after the shell, even during the shelf life, it should be in the same form. It should not become a solid, or it should not absorb moisture. So these are some of the traditional standards that we have to adopt. Next. Now this one thing you can observe. I am showing you some case examples. This is my PhD, one of the PhD drug, where this, palagare, Cypria annulus or Moniteria annulus. This is one variety we get in the market. See, the distinct feature of this particular variety is it has a bright or orange colored ring on the upper side of the shell. Next slide. This is Cypria monita. This is another variety of palagare that is available in the market. And what is given in the text is Cypria monita. When we went for authentication, we showed 2 different varieties, and the zoologist came up with saying that this is only Cypria monita. How do you define it? It has a very thick shell containing an yellow bands across the shell. Can you go to the previous slide? Here, can you make out the difference? The shell contains an annular portion, right? Orange ring. So this is annulus. Next slide. So the shell is thick and it has yellowish band, yellowish discoloration. There are some horizontal bands contained. So this is Cyprium monita. Next. And this is what we come across. Power, of course, to get into authentic power itself, it was very. This is another work of my colleague, Dr. Ratnamala. So, this by a macroscopy, it was very difficult for us to separate the adult trends. We sorted it off with only a microscopic study. Next slide. So this is the microscopic picture. Next slide. Picture. So where I think plastic. No, previous. If it is going to be a plastic material, these striations which are seen in the power by microscopy will not be present. So you could see the depression that is present on the surfaces. Now, these are some of the authentic standards when it comes to power. So in case of power, the microscopy will be a better tool to authenticate the raw material. And of course, where we are getting it from is also very important. Next. So, as far as minerals are concerned, what all the standards that are given in the pharmacopoeia are, they will give you the definition. Where does it? What is the broad classification of that particular mineral? Where does it origin from? As far as minerals are concerned, these physical properties are very, very important. Nature, color, streak, fracture, luster, tenacity, transparency, hardness, and specific gravity. So, if we need a detail of what it is, we need a separate session to know what are all the what does this mean. So, what is the applications of these particular properties? Just remember the physical properties in minerals like nature, color, streak, fracture, luster, tenacity, transparency, hardness are very, very important in authenticating a mineral raw material. Next. Next slide. Coming to the chemical properties, we know that one particular metal or one particular mineral will react with only specific acids. And we also have specific assays. If you take copper as a raw material, then the standard given in the pharmacopoeia is the copper, when you do an assay of copper, it should not be less than 99.5%. Okay, so this is one assay that is going to define your copper raw material. When it comes to heavy metals in copper, the limit of lead is 5 ppm. Like it should not exceed 5 ppm. Limit of arsenic is 1 ppm and cadmium is 5 ppm. Similarly, when you do an assay of other elements, or all the other elements that can be present along with copper is zinc and argentinum. Silver, gold is 135 ppb. These are some of the other things that are present along with copper that is permitted. Next. So this is a thing taken from Ayurvedic Pharmacopoeia of India, Part 1, Volume 7. Copper. So they have given you the definition, what is the synonym, and what are all the other regional names, the picture of copper ore. Next. How is it being, where does it originate from, and what are all the characters? I told you, the nature, either it is seen as wire or how plate, what is the color? It is copper red in color. What is the streak? You take the mineral, take a pinch of it, make a streak. What does the color look like? These are very specific nature that is given for minerals alone. Next. The chemical properties, reaction with acids. As I said, assay, the copper should be not less than 99. So, Shodhana means like how do you use it in a formulation? In us, we have specific purification process. Next. So, when it comes to modern analytical tools Like we have SEM EDAX. EDAX will tell you energy dispersive X-ray analysis. It will tell you the chemical profile, what are all the different elements that are present, what is the percentage of it. Scanning electron microscope will tell you like how the particles look like when it is in a micron level, when it is in nano level or something. How does the particle really look like? Whether it is smooth or rough, what is the shape of the particle? All those things. Similarly, transmission emission microscopy is STEM. That will also be a higher resolution for SEM. That will also tell you how is your particles that are present in your parpam or chenduram or your mineral fine particle. This is going to describe on the minute particle that is present in your drug. Next. XRD is a specific test for the minerals, X-ray diffraction analysis. And there are two different forms in our drug. One is crystalline. Crystalline means any basic mineral or a product of a mineral will contain specific crystal that is repeatable. Or sometimes when it doesn't follow a pattern, we call it as amorphous. So that nature we can understand using XRD. And we also it also has single crystal. So when your drug is really crystalline, we can take one crystal and see how does it look like, what are all the angles that are present, are they similar, everything that we can study using single crystal XRD. Next. And ICP-OES is inductive coupled plasma optical emission spectroscopy. Similarly, ICP-MS is inductive coupled plasma mass spectroscopy. This will give you an entire picture of the what are all the different elements that is present in the raw material or the final product along with its concentration. Coming to infrared, infrared will tell you when you run an infrared, it will tell you what are all the different functional groups. Functional group means whether it contain acid group, OH group, or a CH group, how is the bonding. Everything will be read using infrared. Next. Coming to thermogravimetric analysis. This will, when we subject our drug for higher temperature, we'll know like whether it is going to be, whether the composition is going to be same or it is going to decompose. You will get a graph and you will know whether your product is stable or not. So by this thermogravimetric analysis, we'll come to know the shelf life of that product also. Next. See, I am giving you some of the paper references where they have worked on the standardization process of certain mineral drugs. I'm not going in detail of it due to lack of time. I will just read the topic and what are all the analysis that is done. You can go through the entire paper and see what are all the conclusions that I've made because of those tests, how they have arrived onto the specification for that particular drug. There's one paper that speaks on the process standardization studies on Gandhaga Rasayana. This is done by Brinda et al., published in Internal Journal of Pharmaceutical Sciences. Okay, next. Chemical standardization of Purna Chandrodaya, of course, by one of our Siddha expert who is working on the quality control aspects. And this paper has been a very good model for my team PhD mates who have been working on the mineral and metallic drugs. Here they have done solubility studies. When they went for metallic analysis, iron, then sulfur were present. And by the EDAX studies, they have concluded that mercuric sulfate was a very important component. And also the SEM and DLS study showed that 10% of the particles were below 112 nanometer. So this is the distribution of the particles also. You will know like 20% of the particles are within the nano limit or 30% of the. And one wonderful thing what in these papers what we have observed is only minute like 10 or less than 10% was coming under the nanoparticle range. The other were of macro sizes of microns and other things. So what it shows is if we have all the particles in nano, then we will end up with nanotoxicological things. So our siddhas and the process is so designed in such a way that the entire particle doesn't turn nano as one beautiful observation which you can find out in all these studies. Next. Here, chemical standardization of Rasagandhi Medida. This is done by Sudha V et al. This is also one good study which will tell you on the HPTLC fingerprint of the whole Rasagandhi medicine. GCMS analysis, it has showed like 71 bioactive ingredients, including fatty acids, and what are all the quantity that is present in this particular formulation. Because doing for a raw material, there are n number of papers. They have only limited papers for the formulations to work on. Next. This is a Vanka Parpam. This is one of Dr. Parimala. She has taken Terayar Murai of doing Vanka Parpam. You know, the Terayar Murai, it is not single process. We have to continuously do Puram for 10 or 15 times. That depends on the particular method. So she has studied the entire process, wrote all the chemical changes that has happened in each and every cycle of calcination. So this is really a wonderful paper that everyone can go through. Next. So I think I have ended up. These are some of the insights. Whether you have understood wholly, that I don't know. Whatever that you have come across, or if you find it difficult to understand, you can just contact me in person or through mail so that I can help you out to the knowledge which I have, to the experience which I have learned from my mentors and also from my teachers and also from the laboratory Asagari Herbal Research Foundation where I was working. Of course, my PhD studies and also the work experience which I have gained from Astagiri is what is making me travel in this Konapadam field, both as a teacher and also a researcher. Of course, these are yet to be like, it has to be fine tuned by me, and I myself have to learn a lot. And each and every, now I'm working on one project on Navacharam. It is too challenging even to freeze on the parameters on the raw material parameters. So every study, everything, it's a learning process that we should keep working and join our hands with each other to take up our system in a better way onto a global way where everyone can know about that our science is also well advanced. To my knowledge, this is only my opinion. I would say as though the so much instruments are there to analyze chemically, and as a wholesome, we can say like the parfum contains it is in one particular chemical form. It is not like that. That can be described in the advanced chemistry. So with this, I conclude my lecture. And if there are any interactions that can be made, and if you like to discuss with me, you are welcome. Thank you so much, and thank you for Jignasa and young students who has picked me as one of the lecturers. Thank you so much.
Thank you, ma'am. Shall we move on to doubt clearance session, ma'am?
Yes, of course.
Hello, ma'am. How can in-house formulation prepared by a practitioner can be standardized apart from traditional tools of standardization?
See, and I forgot to tell you one thing. See, though we have pharmacopoeial standard, that standard, this standard, apart from all these, we can also create, even in a pharmacy or a pharmaceutical setup, we can create our own in-house standards. See, for example, if the pharmacopoeia doesn't tell about a product because the pharmacopoeia is not complete, and we have n number of drugs that we can make by ourselves or we can make as a product. What do? I have told you so many analytical tools or analytical techniques. So these can be adopted to your particular product, and we can develop our own in-house standards also. So when it is a practitioner, you want to do it for yourself and for your clinical practice. Apart from the traditional standards, you can just analyze, give it to a lab, get it tested, for one product. There are some specifications that the lab can guide you, and there is PLIM guideline that will tell you. Even in that, we can even pick the essential parameters or not and fix up the standards. That holds good in case of a practitioner. So repeatedly, if you want to check again, also that can be done with a minimal invasive test that we can do with a drug testing laboratory that is accredited by uh, Ayush. So there are 9 to 10 laboratories.
Thank you, ma'am. Next question.
Yes.
How can the preparation of herbal medicines can be standardized when many medicinal plants today are genetically modified or grown in polluted, nutrient depleted environments?
See, to answer this, genetically modified plants are man-made. Or to trace out whether it is genetically modified itself, we need some authentic parameters. So for that, if you adopt these steps, so for example, if you take one plant, of course, for example, if you take neem itself, in Chennai and around areas, you could see lead contamination in neem. Similarly, you can observe a pattern of particular heavy metal contamination in particular drug. So this, unless or otherwise we test and know, then we cannot overcome that. And our concept is, instead of depleting the natural resources, if the plant can be cultivated in a non polluted and a better environment, then that holds good for a good product. So what the major industries do is, even before going for the GMP practices, they adopt good agricultural practices. They take up a land by themselves. They cultivate their own plants or those things. So that would be a better practice. Is it okay for you? Means, are you satisfied with the answer which I have given? So it's better that we have our own agricultural practices also developed for our own pharmacy. That would be a better practice.
Yes, ma'am. What are the steps to get GMP certificate for a Siddha pharmaceutical?
See, as far as we have state licensing authority, we have a separate office for that in Armakam, just opposite to Anna Hospital. The licensing is given by the state licensing authority for all the state. Every state has its own state licensing authority for, especially for ISM products, Indian System of Medicine products. And along with the licensing, they also visit your pharmaceutical setup. They also give a GMP certification. So you have to follow that. If your pharmaceutical is equipped with those facility which has good requirements, so the guidelines and the Drugs and Cosmetic Act will give you certain guidelines. Like preparation of a churnam needs a particular area, like 10 into 10. So for one dosage form, you need a specific area to be manufactured. If you adhere and if you have clear cut steps and if you have submitted the products along with the license or along with the testing, a certificate of analysis, we call it. A particular product, if you get a certificate of analysis with a particular OISH accredited lab, and then they will give you a GMP certificate along with your license. This is how a GMP certificate is got for a Siddha product. So it is given by state licensing authority.
Yes. One last question, ma'am.
Yes.
What is the standardization differences between in-house pharmacy and pharmaceuticals?
See, there is no big difference. Standards and standardization process are same. Let it be even an in-house pharmacy or a pharmaceutical. What will you say? Like, I need a. See, if it is a rose flower, it has its own characters. Whether it is going to bloom in Chennai or it is going to bloom. Of course, the phytochemicals may vary depending on the nature and the season and the altitude. But the standards are not going to differ when you are going to do it as an in-house pharmacy or in a pharmaceutical. And the guidelines or the ministry is going to tell you have to follow certain steps. For a product, these are some mandates. So if they are missing or if you don't get any guidelines or standards, as I told you, with the collaborative thing or with the testing facility, we can tie up and we can develop our own standards saying that a particular product has this much of alcohol content you have to do a 3 batch analysis. It is not sufficient if you do for 1 batch and say it has this much amount of alcohol. It is not like that. You have to manufacture the product 3 times, a minimum of 3 batch analysis. You do a functional group analysis. Like if the first assay says the alcohol content is 9%, the second assay says 8.9%, and third assay says 8.5%, the deviation is 0.25. Okay, so you arrive on to an average and you have to say specify that the product should contain 8.53% plus or minus 2%, something like that. So these are some of the in-house standards that we can develop on our own when the regulatory authorities is not going to give you these standards, because it is highly difficult to stabilize and say we have a number of products for all the products, and you know that the standardization procedure and the pharmacopoeial development is a bit slow process. For one product, if you want to standardize and know the specification, it may take some time. So we need some expertise, we need some laboratory conditions for that too. So in that case, we can simply arrive on to the physical-chemical methods which I was telling into, like pH, the macroscopical characters, like ash content, uh, moisture content, a simple TLC fingerprint, all those things. And as I said, the functional group analysis like total alkaloid content, tannin content are simple tests that we can define our own products in in-house or in pharmaceutical level also. Even in major pharmaceuticals also, they will have their own dossier for that particular product. Dossiers are nothing but the product profile or the specifications that we keep as standards for our own product. So these are the dossiers. So we can develop in-house or in-house standards also based based on, once you fix those standards for every time you manufacture a project, you can just check onto those parameters or not. That is also allowed.
Thank you, ma'am. Thank you so much for your detailed explanation.
And thank you so much that you have given so much time to me and for your very patient hearing. And I am very much glad to share my experience on this topic, especially with the young students.
Yes, ma'am. Let's move on to end of the session, ma'am,
Sure,
with vote of thanks.
sure.
I express my sincere and heartfelt gratitude to Dr. Sudha Revati ma'am for sharing valuable insights on Siddha drugs and standardization. This session was highly informative and enriched our understanding of the subject. Now, I invite Sri Nitish Kumar, State Convener, Jignasa South Tamil Nadu, to present the valedictory speech.
Respected speaker, dear participants, and the Karikatar of Jignasa South Tamil Nadu, a very warm welcome to all of you. As we reach the valedictory section and bring this enriching seminar to a close, I feel a deep sense of satisfaction and gratitude. On the occasion of 9th National Siddha Day 2026, Jignasa South Tamil Nadu organized this webinar series under the theme Siddha for Global Health, with a vision of highlighting the timeless relevance and global potential of Siddha medicine. Over this course of program, we have witnessed the meaningful academic deliberation, insightful clinical discussion, and inspiring perspective rooted in the classical Siddha wisdom and contemporary health needs. I'm happy to share that this program received an overwhelming response with a total of 997 registered participants from the various Siddha institutions, academic background, and professional stages. These remarkable participants reflect the growing interest, responsibility, and the commitment of Siddha fraternity towards the learning, innovation, and global outreach. This webinar series would not have been successful without the invaluable contribution of our esteemed speakers. So I express my gratitude to all the resource persons who generously shared their time, expertise, and their experience. My sincere thanks to Dr. K. Ilavasan sir, a consultant Siddha physician at Ayush Clinic, Southern Railway Headquarters, Chennai, for his insightful clinical perspective and practical approach. I also extend my thanks to Dr. P. Silambu ma'am, Associate Professor, Excel Siddha College and Research Center, for her scholarly presentation and academic clarity. I deeply appreciate Professor Dr. N. Abdul Aziz sir, a founder and director of Marutam Clinics, a renowned Varma therapist and expertise Siddha physician, for his inspiring thoughts, dedication towards the advancement of Siddha medicine. I also thank Dr. K. Madan sir, a consultant at Chittire Chikitsalayam, a Siddha Wellness and Rehabilitation Center, for sharing his valuable insight drawn from the clinical practice and community medicine. My heartfelt gratitude, Dr. S. Sudha Revati ma'am, Assistant Professor, Department of Gunapadam, National Institute of Siddha, Chennai, for her scientific and academic contribution that enriches the section. Each speaker added a unique dimension to this webinar series and collectively reinforcing the strength, depth, and the global relevance of Siddha medicine. I also take this opportunity to appreciate the dedicated efforts of the Jignasa South Tamil Nadu Organizing Committee, coordinators, technical team, and the volunteers whose meticulous planning and commitment ensured that the smooth and successful execution of this program. Dear participants, your active involvement, attentive listening, thoughtful interaction made this webinar series a truly meaningful one. Your enthusiasm assures us that the future of Siddha medicine is bright and in the capable hands. As we conclude, let us take forward the essence of this program to preserve the classical Siddha wisdom, to practice with scientific integrity, and also to contribute towards the positioning of Siddha medicine as a global healthcare system. I am confident that the knowledge gained through this webinar series will support you in your academic growth, clinical excellence, and professional journey. With these words, I formally conclude the webinar series of 9th National Siddha Day organized by Jignasa South Tamil Nadu. Thank you once again for your valuable support and participation. Thank you.
I express my heartfelt gratitude to all our esteemed resource persons who over the past 5 days have generously shared their knowledge and experience, making this academic webinar series on Siddha for global health on the occasion of 9th Siddha Day truly enriching and meaningful. I extend my sincere thanks to the organizing committee for their dedicated efforts, careful planning, and consistent coordination throughout the 5 days, which ensured the smooth and successful conduct of this program. Once again, I thank everyone who contributed directly or indirectly to the successful conduct of this webinar. With this, I conclude the session. Thank you all.